Hello girls. Now in this video lecture we will discuss about demonstration of sugar fermentation. So what do you mean by fermentation? It is a biochemical pathway which uses specific enzymes for specific sugars through fermentative pathway. What do you mean by this? As a result of fermentation, certain end products are being formed by the bacteria. The bacteria, when it is being growing in any media which consists of any source of carbohydrate like glucose, maltose, sucrose, or lactose, it utilizes that particular carbohydrate, and as a result it is able to ferment it. That means it is able to metabolize it and produce certain end products. These end products are acids and gases. Some organic compounds are being produced by that particular microorganism when it is utilizing a particular carbohydrate source like sucrose, lactose, glucose, maltose, whatever is the source of carbohydrate which is being present in the media with that particular bacteria is growing. Now here in this demonstration of sugar fermentation, the aim of the experiment is to test for a given bacterial organism for its ability to ferment a given sugar. Whether or not a particular bacteria is able to ferment or metabolize that particular sugar, and as a result, as if it is able to produce or ferment the carbohydrate, it should produce certain end products. We can detect and demonstrate that particular end products which are being produced by that particular microorganism. That can be done by this by performing this particular experiment, that is sugar fermentation or carbohydrate fermentation. Now the main principle lying behind this experiment is, now bacteria will use variety of carbohydrates as a source of energy like starch, which is a polysaccharide, lactose, maltose, sucrose, glucose, fructose. Okay, many polysaccharides, disaccharides and monosaccharides which are mentioned here are being used as a source of energy by bacteria. Now they will ferment these sugars or they will hydrolyze the sugars, and as a result they ferment and as a result produce certain end products like acids and gases. These can be detected, that is acids can be detected by using a pH indicator. It may be phenol red, neutral red or methyl red. Any of the indicator, pH indicator can be used to detect the presence of acid, whether or not the bacteria has utilized or fermented the sugar or carbohydrate in the medium, and if it has produced acid, that can be detected by using a pH indicator. Now, gas production is also been detected by using a small tube which is being inserted into the test tube, normal test tube with the broth, and as a result gas is being produced or collected in the Durham's tubes. Now, what are the requirements? The requirements include lactose broth. The media which we are using is lactose broth, and the culture or the test organism against which we are going to demonstrate this sugar fermentation is E. coli. We are using the bacteria E. coli, and methyl red indicator. Here the pH indicator to test for the presence of acid, we are using methyl red indicator. Coming to the procedure, so the test culture is inoculated in sterile lactose broth tubes. So we are taking test tubes with lactose broth in it, and then we are inoculating the test culture. What is the test culture here? It is E. coli. That means we are simply inoculating the lactose broth tubes with E. coli organism, and after inoculation they are incubated at 37 degrees Celsius for 24 hours in the incubator. Now that means if E. coli is able to ferment or utilize the lactose, that means if fermentation has occurred, as a result of fermentation, end products are formed. What are those end products? Organic acids and gas. Now we will look for the formation of a gas bubble in Durham's tube. as I mentioned that it's a small tube which is being present or which is being kept inside the normal test tube with the lactose broth. If gas is produced, a small gas bubble is formed in the Durham's tube. And now for the detection or presence of acid is been produced or not, we are going to add methyl red indicator to the presence for checking the presence of the acid production. Now, after adding this methyl red indicator, a red color is being developed or produced which will indicate acid production, and presence of gas bubble will indicate the gas production. Let us look into the pictures and then discuss. So this is exactly what is a Durham tube. So here this is what is called as the Durham's tube, the small tube which is present, which is being kept inserted in the normal test tube. This is the normal test tube with the broth, and this is the gas here. This bubble, whatever is there, this is the gas. And here it is showing that it is acid. Usually you can add acids, they get accumulated at the top, and you can add the indicator and you can see the change in the color. So this is the Durham's tube. This is the small Durham's tube, and this is the gas bubble. Now when we perform this test, so here on the right side this is a positive, so it is indicating plus and minus. It is a positive test and this is the negative test. Why it is positive? This is positive for acid production. If acid is produced when you add methyl red, that means acid at 4.2 and below 4.2, that is at lower pH, the color of the methyl red will remain red in color. Though the color of the methyl red is red in color, that means at lower pH, the color will turn into red or pink in color, which indicates that it is a positive result or acids are being produced. But at higher pH, even though you add methyl red, it will not turn red in color. If it is alkaline in nature, then it will be some yellow in color or some other color. It will not turn into red color. Then it indicates that acid is not produced. This is in case of methyl red. This can also be performed, this sugar test can also be performed with other indicators like phenol red indicator. Now this is an example when we are using phenol red indicator. This is added prior. So here when we are talking about this methyl red, this is been added after incubation, methyl red. But here in this case phenol red, we are adding prior, that is along with before inoculation with E. coli organism itself, we are adding phenol red into the broth and then we are inoculating with E. coli and then incubating. So what is happening as a result of fermentation? Already phenol red is present in the broth. E. coli is also inoculated and E. coli will utilize the lactose broth, and as a result it will ferment and produce acids and gases. So here it is giving a positive result. So this one is a positive result for both acid and gas. For acid and gas, it is giving a positive result. So this is indicated as gas which has been present in the Durham's tube, and a yellow color. So when you add phenol red, when we are using phenol red for the indication of presence of acid and gas, sorry, this is for not gas detection, only for detection of acids we use this indicators, and it will turn yellow in color. Yellow color. So yellow color, turning the broth into yellow color. That means phenol red at lower pH, it is yellow in color. At higher pH, above 7 and alkaline nature, it is red to pink in color. So this is in case of phenol red. So it is yellow in color at lower pH, that is around 4.5 like that. If acids are accumulated, so the phenol red which is already present in the broth, because of the presence or accumulation of the acids, it will turn yellow in color, and this is a positive result for both acid and gas. So this is a negative result. So if there is no formation of acids and gases, this is how it gives a negative result. This is with case of same phenol red again. So this is a positive result for acid production, because there is no gas produced in the bubble Durham's tube. So this is both acid and gas positive result. So this is about the sugar fermentation test. So this is how we demonstrate in laboratory by adding certain reagents, especially for detection of the acid, that is methyl red or phenol red is been added into the medium, and change in the color will give us whether it is positive or negative. That means whether the organism has utilized the sugar or the carbohydrate, and whether it has performed the fermentation. So this is about the demonstration of sugar fermentation or carbohydrate fermentation. Thank you.